Welcome to video tutorial of Nigga Academy. Today we shall see RNA. Last time in my video we tried to discuss nucleic acid, particularly DNA. DNA is deoxyribonucleic acid. The objective of today's lesson is to see structure and function of RNA. We know that DNA is a double strand. DNA is a genetic material. What about RNA? Is it double strand? Is it a genetic material? We will see. What are different classes of RNA? What is the function of each class? Let us see. RNA is ribonucleic acid. It is present in all living cells. This rRNA, unlike DNA, it is a single strand. As you can see from this diagram, it is a single strand with alternating phosphate and sugar backbone. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Its backbone is made up of sugar and phosphate. Sugar and phosphate. Mind you, unlike DNA, it is a single-stranded. The nitrogenous bases on RNA are uracil, cytosine, adenine, and guanine. Mind you here, this nitrogenous base is connected to sugar by glycosidic bond, just like DNA. Here is 5-carbon sugar, which is ribose. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. It is ribose because there is free hydroxyl group at the carbon number 2. Nitrogenous bases found in RNA are uracil instead of thiamine. The phosphate group attached at the carbon number 5 and at carbon number 3. The direction of synthesis is from 5 to 3 direction, just like DNA. Look at here. Carbon number 1, carbon number 2, carbon number 3. So new phosphate will be attached at the carbon number 3 here. Then around this, here is carboxyl group here. It will be attached with hydroxyl group, carboxyl group of sugar. Then the coming phosphate could be attached at the carbon number 3. In such a way, RNA synthesized. I will come later on this issue. RNA synthesized upon DNA template. It cannot stand by itself like DNA. It made upon existing DNA template. Mind you, it is a single-stranded. But it may form double strand, for example, during secondary structure. You know, RNA is a single strand. On that single strand, there is a sequence of nitrogenous bases. There is a sequence of nucleotides. In that large sequence, complementarity may happen. When I say complementarity, the nitrogenous bases that can pair with each other. Look at this diagram, this figure. Adenine, cytosine, guanine, uracil. Adenine, guanine, cytosine, like this. Then it turns upon itself. Then it forms complementarity. Here is cytosine, here is guanine. Here is guanine, here is cytosine. Here is adenine, here is uracil. Here is adenine, here is uracil. So it forms complementarity. Mind you here, it is upon a single strand. It forms a complementarity. during secondary structure formation. RNA do have primary structure, secondary structure, and tertiary structure. So during secondary structure, there is pairing of complementary bases. Adenine pair with uracil, cytosine pair with guanine. So in such a case, it forms double strand. There is hairpin, loop, like structure could be formed. What are different classes of RNA? In generally, RNA can be coding RNA, non-coding RNA, and this non-coding RNA again, there is regulatory RNA. When I say coding RNA, typical example is messenger RNA. The other ribosomal RNA, transfer RNA are non-coding RNA. Non-coding RNA. The others are regulatory RNA. Actually, they are non-coding. They are regulatory. They regulate the process of transcription and other gene expression processes. These are regulatory RNA. Let us see coding RNA, which is a messenger RNA. You know that this messenger RNA is formed upon DNA template. Suppose this is a nucleus. Nucleus is found in cytoplasm. In the nucleus, what will happen? Replication of DNA carried out. You can see my video of DNA replication. After replication, transcription will begin. When I say transcription, the information contained in DNA can be converted into that of RNA. Mind you, in DNA, there is a thymine. In RNA, there is uracil. The rest are the same anyway. So the sequence written in the language of DNA can be converted into that of messenger RNA. It means that messenger RNA is formed upon existing template strand of DNA. Once pre-messenger RNA is formed, it could be processed with help of regulatory RNA. After that, it moves to the cytoplasm, where protein synthesizing machinery is found, particularly ribosome. Here on the ribosome, protein synthesis could be carried out. There are so many things. Different ribosomal RNAs, transfer RNAs are there. There is initiator proteins. So with help of that entities, protein synthesis could be carried out on the surface of ribosome. So bear in mind that messenger RNA, it carries gene information and then move to the protein synthesizing machinery. That is the point. Look, this is a DNA strand. One of DNA strand serves as a template. The other serves as a coding strand. RNA synthesized upon DNA template strand. Look at here. Here is cytosine, guanine will be produced. Here is thymine, adenine could be produced. Here is adenine, uracil could be produced because there is no thymine in RNA. Here is cytosine, guanine could be produced. Here is guanine, cytosine could be produced. Bear in mind that RNA, messenger RNA is synthesized upon DNA template. One of DNA strand serves as a template, the other serves as a coding strand. This positive one is a coding strand. It is a sense strand. So RNA synthesized upon DNA template. Its sequences complementary to the DNA template strand sequences, but it is exactly similar to that of coding DNA strand, except thymine and uracil. So the sequence carried on messenger RNA is read in the form of codons. Codon is nothing, it is a triplet, 3 nucleotides. Look, for example, guanine adenine uracil, one codon, it may code certain amino acid. Guanine cytosine adenine, another codon, it may code another amino acid. Guanine uracil guanine, this also another codon, it may code another amino acid. In such a way, it codes those different amino acids. When you see the structure of this messenger RNA, at the 5' there is what we call 7-methyl guanosine triphosphate cape. Here is a cape. Around this, there is a cape. Around 5' there is polyadenylation. So many adenine, around 20 to 250 sequence of adenine. So, this cape at the 5' is very important. 1, for the protection. 2, for the recognition. The same is true for this found at the 3'. Look, for example, you know RNA is synthesized in the nucleus. The environment in the nucleus is different from that of cytoplasm. In the cytoplasm, there are so many enzymes that can degrade RNA. In such a case, this RNA could be produced and processed, mature form of messenger RNA moved. When I say mature form, capping will be made at the 5'. Polyaddition could be made at the 3'. So they are very important for the protection from different enzymes. The other importance of that cap is for the recognition. You know, as I said, on the surface of ribosome, there is different ribosomal RNA, transfer RNA, initiator proteins. So those initiator proteins recognize the codons carried by messenger RNA. Messenger RNA, especially during protein synthesis. Anticodons found on transfer RNA easily recognize codons on messenger RNA. That may be due to that cape. So that is the importance of the cape which is found at the both end. The other group is transfer RNA. As we can see from its name, it transfers amino acids from cytoplasm to the protein, synthesizing machinery. So its name is transfer RNA. It transfers amino acid. This molecule is called adapter molecules because it serves as a link between messenger RNA and amino acid sequence. It serves as a bridge. It carries codon sequence and then moves to the protein synthesizing machinery where proteins can be synthesized with the help of transfer RNA, with the help of ribosome. So it serves as a link. It serves as a link. You know, there are 20 amino acid. There are 20 transfer RNA for each amino acid. For each amino acid. 20 species of transfer RNA, 20 amino acid. It provides amino acid corresponding to each codon. Each codon. That is the primary role of transfer RNA. Look, this is a messenger RNA. Messenger RNA carries codon sequence. Transfer RNA do have anticodon arm. That anticodon arm recognize codons carried by messenger RNA. Then it recognizes that sequence. There is an enzyme. That enzyme aid in adding responsible amino acid for that codons. For example, UUU. This is anticodon. So lysine could be added. This is CUA. ASP protein could be added. The point here is protein synthesized on the surface of ribosome. Messenger RNA carries code sequence. Then transfer RNA do have anticodon arm. There is anticodons. they recognize that sequence and they add responsible amino acid, responsible peptides. So it forms a polypeptide. That is the point. When you see the structural characteristics of transfer RNA, it can be primary, secondary, and tertiary structure. When I say primary, it is a kind of sequences that may generate secondary structure. Secondary structure. Very long sequence. That is a primary structure. Secondary structure, it forms a double helix. It forms something, a double strand. Because there could be extensive internal base pairing because of complementarity on that single sequence. When that secondary structure combined, folded, it forms tertiary structure. It seems like clover leaf due to hydrogen bonds between T and Dr. T and Dr. This T and Dr. So it forms tertiary structure. Look at secondary structure here. Messenger RNA carries codons and come to the ribosome. Then here is a transfer RNA. Transfer RNA do have anticodons that can correspond to that codon. This region is anticodon arm. It is important for the recognition of codons carried by messenger RNA. Here is a DHU arm. This is a site for an enzyme amino acid transfer RNA synthesis, which add peptides, amino acids, to the acceptor arm. So amino acid transfer RNA synthesis enzyme add amino acids to the acceptor arm. Because at acceptor arm, here is 3' A, C, like sequence. So here, there is a connection of carboxyl group of amino acid with that of free hydroxyl group, which is found at the 3'. At the 3'. There is a carboxyl group on amino acid. There is free hydroxyl group at the 3' of this sequence. So they connect, that they can be added. Mind you, this amino acid transfer RNA synthesis add responsible amino acid at the 3'. What the role of tR? It involves in binding of transfer RNA to ribosome. Look carefully. On the ribosome, proteins can be synthesized with the help of different bodies, like transfer RNA, like ribosomal RNA, like messenger RNA. In such case, this arm, T arm, help in binding of transfer RNA to ribosome. That's the role of this arm. That's the role of this arm. What about ribosomal RNA? Actually, this ribosomal RNA is much more in number, but its role is maybe structural role. Sorry, maybe structural role. You know, in mammalian, there is ATS that can be large subunit and small subunit. Larger subunit, 60S, this is nothing but molecular weight. 60S and 40S. 60S can be 5.8, 28S, maybe 50 polypeptides. Small subunit, maybe 18S, maybe 30 polypeptides. In bacteria, there is 70S ribosome. Larger subunit 50S, smaller subunit 30S. 50S can be 23S, 5s subunit, 30s 16s subunit. Actually, this 16s is very important for research purpose nowadays because 16s ribosomal RNA do have conserved region and hypervariable region. When I say conserved region, the region which is similar throughout generation, from generation to generation, it is similar. When I say hypervariable region, difference between species. There is V1 up to V4, hypervariable region, maybe in bioinformatics. So anyway, there is both conserved and hypervariable region. So this is very important to study phylogenetic evolutionary relationship. So it is very important. The function of ribosomal RNA is very important in ribosomal assembly. It seems to play key role in binding of messenger RNA to ribosome and may be important in process of translation. For example, in case of bacteria, 16s ribosomal RNA do have child organo sequence. There is a unique sequence which is child organo sequence that is very important to initiate the process of protein synthesis. The other role is peptidyl transferase activity. Ribosome do have 3 sites, P site, E site and A site. P site is the site by which peptide could be added. A site is entrance, E site is exit. P site is the site where peptide will be added. That may be the role of ribosomal RNA. Because that reason, it performs peptidyl transferase activity. The rest are non-coding and they are very important in regulation process. They are called regulatory RNA. For example, microRNA do have 22 to 24 nucleotide long. Its function is silencing and post-transcriptional regulation of gene expression. It regulates gene expression by base pairing with complementary sequences in target messenger RNA. This may lead to degradation of messenger RNA. That is the role of microRNA. It's very important in RNA silencing. Again, in post-transcriptional regulation. After transcription, post-transcription could happen because immediately after transcription, pre-messenger RNA will be produced. That pre-messenger RNA cannot perform its activity because it is immature. Because of that reason, that pre-messenger RNA should be processed to produce mature form of RNA, messenger RNA. So that is the role of microRNA. The other group is small interfering RNA, around 20 to 24 nucleotide long, like this. It is very important also in post-transcriptional regulation of gene expression. Sometimes it may clear or degrade messenger RNA that no longer needed by cell or clear viral RNA. So bear in mind that after transcription, there should be post-transcriptional regulation. You may see the detail of this in my other video. Small nuclear RNA, it is a long 150 nucleotide. The same is true for processing of pre-messenger RNA, regulation of transcription factors. There are so many transcription factors. It regulates, maintain telomeres on chromosome, telomeres. The other long non-coding RNA is very long, around 200 nucleotide long. It is very important in chromatin modifications. Again, the regulation of transcription, splicing. When I say splicing, removal of introns connection of exon. Introns are nothing. They are non-coding region. They code for nothing. They should be removed. So that is what you call a silence. In such a process, this long non-coding RNA is very, very important. The other group is small nucleolar RNA, around 82,000 nucleotides. Very important in chemical modification of other RNAs. Ribosomal RNA, transfer RNA, small nuclear RNA. As a summary, RNA contains ribose sugar and nitrogenous bases, adenine, guanine, cytosine, and uracil. But DNA is deoxyribose. RNA is ribose. No thymine here. Instead, there is uracil. DNA is single strand, but may form double strand during secondary structure formation because of complementarity. RNA is not genetic material except in some viruses. Rather, it is very important in the process of protein synthesis. Different classes of RNA, messenger RNA. This messenger RNA is a coding RNA because it codes for proteins. It codes for the amino acid. It contains a sequences. Ribosomal RNA, it's very important in ribosomal assembly. Do have structural role. Transfer RNA do have many role in protein synthesis. It recognizes codons carried by messenger RNA. It adds new amino acid. It helps in binding of transfer RNA. Anyway, it's very important in the process of protein synthesis. The rest RNA are regulatory RNA. These are regulatory RNA. Thank you for watching this video. Subscribe, share and comment on this video. Thank you very much.